Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, Stitcher, Pandora, TuneIn Radio, GoodPods. Whatever you listen to podcasts, you can find Let's Talk Micro. As for our social media, I am on Instagram as Let's Talk Micro, no apostrophe, on Twitter as Let's Talk Micro 1, on TikTok as Let's Talk Micro, YouTube channel Let's Talk Micro, and on LinkedIn as Luis Plaza. So please go ahead and subscribe to the podcast, download episodes, and leave a review if the app allows you to do so. Definitely any feedback, any suggestions, like any topic suggestions, you can email those at letstalkmicro@outlook.com. But any feedback, any suggestions, they are always welcome and appreciated. And as always, if you haven't listened to the last episode of Let's Talk Micro, the previous one, please go ahead and do so. It was a great episode about extensively drug-resistant Shigella or XDR Shigella, and two experts from the CDC, they came to the podcast, Doctors Naima Logan and Luis Francois Watkins, and they joined the podcast and they talked about Shigella, what makes it extensively drug-resistant, what population is at risk. So overall, it was a great episode, very informative, and on the show notes, there are some resources that you can access and get more information about Shigella. So definitely check it out if you haven't already. And once again, shout out to the CDC because, they have responded so quickly when I reach out for questions. When I reach out for a request with an episode, they, responded very quickly and we got something on the books very fast. So thank you for that. And the audience thanks you as well. So today's episode is definitely in the ballpark of us medical laboratory scientists and microbiologists. This episode is about Piperacillin-Tazobactam or TZP. And those of us that work in clinical micro, we are definitely familiar with it. We definitely do, right? We do susceptibility testing in some hospitals. If this automated system terminates it, we go ahead and provide an e-test. We provide a Kirby-Bauer. It's a drug, it's an IV drug. And if you're patient, it's an inpatient, your policy in the hospital might be to make sure that we provide that susceptibility report on Pip-Tazo. So this is a great episode. And in it, two guests, joined the podcast, which are Dr. Romney Humphries and Dr. April Abbott. Two microbiologists, very experienced, very involved in the field of micro, in clinical micro. You see their names on the CLSI. So they are very active in the community. So definitely, I was very thankful that they had they took the time to join Let's Talk Micro and talked about a study that they did where Piperacillin-Tazobactam was evaluated using automated instruments like your Vitek 2, your Phoenix, your Microscan, and the results were evaluated against a reference method, which is a broad microdilution. And the results were compared. So it was a very interesting study. And in the podcast, they talk about TCP. They talked about the MERINO trial, which is something that I learned about. I wasn't aware of it. So how they were in a trial where they tested TCP and see how it performed against the carbapenem. They were trying to test that it was better, but it ended up being worse. And then one thing that was detected was that some of the automated systems were overcalling susceptibility. So it was being called susceptible where it was not. So definitely a great information in this episode. They talked about how TCP is a tough drug to test. They discussed the study with the errors, how the instruments perform, the recommendations, and a great topic that maybe some of you might not be aware of, which is the clinical breakpoints. What kind of breakpoints do the automated instruments use, which are the FDA breakpoints, and how do those compare to the CLSI breakpoints? So definitely great conversation, great episode, very informative. So let's go ahead and listen to it. So those of us that, that we are medical lab scientists and we work in clinical micro, we're definitely familiar with Piperacillin-Tazobactam or TCP. And I found this, this study, and I thought it would be to share it with the audience, MLS and microbiologists. So the study, is titled Evaluation of Piperacillin-Tazobactam Testing against Enterobacterialis by the Phoenix, Microscan, and Vitek 2 tests using updated clinical and laboratory standard institute breakpoints. This was published in the Journal of Clinical Microbiology in January of this year and by the American Society for Microbiology. So with me today, I have two guests, Dr. Romney Humphries and Dr. April Abbott. Doctors, welcome to Let's Talk Micro.
Thank you. Pleasure to be here.
Thank you as well.
My pleasure. So let's go ahead and just for the audience, can we start with a quick introduction?
Sure. I'm Romney Humphries. I'm the Division Director for Lab Medicine at Vanderbilt University Medical Center. I'm also a clinical microbiologist. So when I get the chance to go into the lab, I'm super happy to see the plates and look at the bugs. And I also do a lot of research on antimicrobial resistance and susceptibility testing.
And I am Dr. April Abbott, and I work at Deaconess Hospital. We're a health system in Indiana, Illinois, and Kentucky. There I direct the micro lab in our molecular diagnostics section for infectious diseases. Have spent the last few years doing mainly COVID-related stuff, but happy to get back into the clinical lab and sort of where my heart belongs with susceptibility testing. So I'm just really excited to kind of be here and talk about the study.
Definitely, isn't like great, plate reading, and it's wonderful, what we do. It's great. And I have seen your names in different publications, so I know you definitely are very active. So, once again, I appreciate the time that you're taking to talk about this. So let's go ahead and talk about the study. Can we do like an overview of it?
Sure. I can kick us off. So this was a study to take a look at what the lab experience might be if they were to update their test systems with the new CLSI breakpoints for Piperacillin-Tazobactam. So we looked at the Enterobacterialis, and so many people in the audience may be aware that CLSI updated the breakpoints for this drug about a year ago. However, up until very recently, the US FDA has not recognized those breakpoints. And so if clinical labs want to adopt them, they have to do a validation study. And we all kind of struggle with these in our labs. It can be really tough to get reference testing done. It can be really tough to kind of know how to evaluate the data. And so we thought it would be valuable to sort of talk about the experience that a bunch of labs had had using these different systems.
Okay. Yeah, it can be definitely challenging sometimes, where to start, what to do. And so definitely a lot of variables here. So, as I was reading the article, it talks about the MERINO trial. Just can you talk about what that is, what that trial is?
Yeah, I'll take that one. So the MERINO trial is a randomized controlled trial that was done across multiple countries, and it was led by David Patterson's group, and Patrick Harrison was the first author. And so this group of investigators were really interested to see if the old sort of standard of going to a carbapenem for treatment of ESBL, E. coli, or Klebsiella was really true, or if from a stewardship perspective, you could just use Piperacillin-Tazobactam. And that's kind of what they were hoping to find, that they could spare the carbapenems and use Pip-Tazo. So they randomized patients across multiple countries who had bloodstream infections, so serious infections caused by E. coli or Klebsiella pneumoniae. And the enrollment criteria were isolates that were non-susceptible to ceftriaxone, so intermediate or resistant to ceftriaxone using current breakpoints. And so then patients either got Piperacillin-Tazobactam or they got meropenem, and they compared the outcomes. The primary thing they were looking at was mortality. So sort of like the worst outcome would be that you are no longer alive at the end of your treatment course or at 30 days. And so as they were enrolling patients, what they found kind of before the trial even finished was that they were not able to show that Piperacillin-Tazobactam was non-inferior to meropenem. It's kind of a funny way that they talk about it, but essentially it means Piperacillin-Tazobactam wasn't as good as meropenem in the trial. And so they sort of went about hoping to prove one thing, and they proved the exact opposite thing, which in science is sometimes kind of nice because, probably your biases didn't play a huge role in that. And so the challenge, though, from this trial, when they went back and did a lot of work trying to understand what had happened, because keep in mind, all these bugs were susceptible to Piperacillin-Tazobactam. But when they went back and reevaluated the data, what they found is that some of the commercial systems that the clinical labs were using, and some of them were also using disk diffusion, were undercalling resistance to these bugs. And so they'd enrolled the patient in the study thinking that they had a susceptible isolate, but when they went back and tested it by reference methods, it was actually resistant. And so it kind of also sort of roundabout proved another point, which is how important the AST results that we generate are to patient outcomes, right? So if we're making mistakes in our labs, that can have a real impact on the patient's outcome.
And I think, another thing that was kind of going on simultaneously around this time is there was really a lot of literature that was starting to come out about how we utilize dosing strategies in the hospital, and those may not be how the original dosing strategies for a particular agent were designed whenever it went through the FDA approval process. So an example here would be Pip/Tazo, where we've seen a lot of literature come out that shows if you're using sort of this dosing strategy where you're giving a single dose and then you're waiting some time period and giving another dose, that you're not probably going to achieve the PK/PD targets that you would need to overcome some of these higher MICs. And so a lot of hospitals have gone to sort of this continuous dosing strategy. And so although that's happening in the real world, it isn't necessarily something that's widely standardized. And so that kind of played a part in this too, where we look at are the MICs that you're getting with the dosing that you're probably going to use in these different clinical scenarios, do those two things go together well? So are you able to achieve those PK/PD targets that you think you're going to overcome that MIC to end up calling it susceptible? So I think all of these pieces of data kind of came together at the same time.
Okay. And so, as I was also reading, and Dr. Humphries, you talk about, you mentioned disk diffusion, definitely a method that is widely used in the lab. So, almost everyone, they use disk diffusion, and, so it says, that it's associated with high rates of minor errors. Can you talk more about this?
Yeah. So when CLSI goes about resetting breakpoints, MIC breakpoints, we also have the chore of making sure that the disk diffusion correlates match those new breakpoints. And so this is getting trickier and trickier, I will say, because we're starting to move breakpoints closer and closer to where the wild type MICs are. And so when the majority of your bacterial population sits right at the breakpoint, there is not a lot of wiggle room for small testing, inaccuracies to come into play. So even though we've, done a lot of work to standardize our disk diffusions, we're all very good about using, the 0.5 McFarland and setting them up in time and incubating the same time. There's still, ranges for all of those things. And all of those, when they add up, could add some inaccuracies to the test. And so when we re-evaluated the Pip/Tazo disk diffusion breakpoints, I think, well, first off, it's important to know we didn't have any really good contemporary data. So most of the data we used to set those breakpoints was from either when Pip/Tazo first came to market, which was in like 1997, and a small study that was done in 2003 looking at ESBL producing isolates. So like the types of bugs that we see circulating these days weren't included in the evaluation just because we didn't have that data. So when we did the best we could and set these breakpoints, it was imperfect, right? And so there were a pretty high number of minor errors, so overcalling resistance by the disk. And then we also saw some very major errors. It was so close to hitting acceptance criteria, but I think the very major error rate was like 2.4, 2.5, and 2 is what you want, right? So it was a little bit higher than what you would hope to see. So part of the problem with the disk is, again, it was designed way back when Pip/Tazo first came to market, when all the bugs were super duper susceptible. And so the disk mass is probably not quite right. So that 100 micrograms of piperacillin and 10 micrograms of tazobactam that's in that paper disk is probably not the optimal concentration to help you differentiate susceptible from resistant in contemporary isolates, and then also using these current breakpoints. So CLSI is working with UCAS to see if we can come up with a better disc content. In Europe, they actually have a different pip-tazo disc than we do. They have a 30-20 disc, I think. No, sorry, 36 disc, around that. And so their disc is different. So they did go through this exercise, but they're still not super duper happy with it. So it's kind of an international challenge that we all face. So yeah, when you're doing disc diffusion, you can expect those results to maybe not quite correlate very well with your automated AST systems, or even with reference broth microdilution.
Okay. So let's go ahead and talk about the testing. So, it's in the article. So about, like how many isolates, and it mentions, what methods, what instruments were used.
Yeah, so I'll take this part. So how this kind of came about, we were having a discussion about how you would do your own off-label validation in your laboratory, right? Because each one of us are really kind of facing the same sorts of struggles here when we have a CLSI breakpoint change. Whereas our instrument is maybe using an FDA-approved breakpoint, or we need to update that breakpoint in the laboratory. So how this really was set up is for each site to collect their own contemporary isolates, and really to try to get certain organisms that we would routinely see in the laboratory, do the testing at your own facility, and then send that off to a laboratory, a single laboratory, which ended up being Vanderbilt, to then do the broth microdilution. So you have to keep in mind as you read through the study, it's not that we were coming up with a study set and sending that amongst the different laboratories. We really were doing this more like we would do our own internal validation for this particular breakpoint change. And so there were 5 different clinical testing sites in which we gathered our own, and we looked at all 3 of the major automated AST systems. So overall, it was about 284 isolates. And some systems, just as it happened, had more isolates than others. And so some of the sites maybe had limitations in terms of gathering those isolates. So you'll see through the study that it wasn't necessarily paired to have the same number of isolates with each particular testing system, but they ultimately all did have those isolates tested in-house and then sent to the same laboratory for broth microdilution.
Okay, and definitely, like the article mentions, so definitely it's a microscan. So the results from the labs that send their isolates, so they were tested by automated systems, like the Phoenix, the Vitek 2 microscan.
Yeah, and I probably should have stated that. So yeah, so microscan, Phoenix, and Vitek 2. And really, we utilize our systems. We do our own internal QC. We follow our own SOPs. Just like any clinical laboratory would, and allowed the system to call those MICs as it would and interpret those. So really there wasn't a manual interpretation done or overreading of the panels. We relied on our systems as they are used in the clinical laboratory.
So the testing is done. So let's talk about the results. So overall, how did the instruments perform and how did they compare to the reference method?
So I think one thing to comment on before we dive into the commercial systems is that pip-tazo is a tough drug to test. It's probably the most difficult drug we test. And this is for a couple of reasons. One reason is you have piperacillin, which is essentially just a penicillin, and you're requiring tazobactam to protect that penicillin against all these different enzymes that these bugs can make. So the inoculum really can have a big impact on where your results might shift a little bit. And so one of the caveats of our trial design is we weren't testing the exact same inoculum by the reference method as we did by the automated systems, which is what you would do in a clinical lab, right? You're not going to have that reference method sitting at your side when you're trying to do your validation. So it was a little bit pragmatic, very real world. The other thing I'd say before we dive into the commercial systems is we did a study prior to this one where we compared broth microdilution to itself. And so we got 2 different brands of piperacillin powder, 2 different brands of tazobactam powder, 2 different brands of Mueller-Hinton broth, and we mixed and matched those to come up with 6 different formulations of broth microdilution. And broth microdilution against itself also did not meet what you would hope to see for a commercial method. And so even when you compare the reference to itself, again, because Pip/Tazo is such a hard drug to test, there were minor errors. There were actually quite a few minor errors, and the categorical agreement was less than what you would hope to see. So putting that in context, right, if we look at the commercial systems, they overall did not hit, like that 90% categorical or essential agreement that M52 says that you should have, which is that CLSI document that talks about verification studies, nor what the FDA would require if this was like a clinical trial data set that was being submitted to the FDA. And so there were some like nuances in the types of errors that we saw. And so I don't know, April, if you want to kind of go over some of that.
Sure. Yeah, I can hit a few of these. So I think, to talk first about, as Romney pointed out here, it really wasn't set up to do a head-to-head comparison. So you can't really interpret, was one instrument performance maybe better than another, or they weren't using the same isolate. So it really wasn't set up that particular way. So, whenever we look at, as an example, the Phoenix here as a whole, the essential agreement was about 83%. So as Romney pointed out, that's a little short of that 90% that we would like to see. And overall, it had a categorical agreement of about 84%. It had a very major error calling something susceptible. So again, remember, a very major error is one where whenever you're doing your testing, you see that it tests susceptible, but your reference method is calling that resistant. So we consider a very major error to be one that you want to try to avoid, because that could lead to treatment of the patient with that drug when it probably is not truly susceptible. And so, we really shoot for, less than 1% or 1% of those types of errors. So when we had these low numbers of isolates that kind of were resistant in that category, then you can see that you're going to have maybe wild swings in terms of what you get for your very major error rates, just as an example here. And that would be true, again, like when you're doing this testing, this verification or validation testing in the laboratory. And so, we typically are going to want to keep those major errors pretty low. And so with this particular system, the major errors were pretty good, less than 2%. The minor errors were above that kind of 10% that we're looking for. But again, it wasn't terrible in terms of what we see. And as Romney pointed out, we still saw some errors even with the broth microdilution against itself. So those are kind of to be potentially expected with Pip/Tazo. So I don't know, Romney, if you want to hit the next one here.
Sure. Yeah. So the other system we looked at was Microscan. We saw a couple more struggles. So I'd say the essential and categorical agreement were very, very similar compared to what we saw on the BD Phoenix. And so 83.6% essential agreement and 80% by the CLSI breakpoints categorical agreement. The thing that I guess kind of stood out was that there were 36% very major errors by the Microscan system. Now, there's some caveats to this, right? There were only 11 resistant isolates, and all 4 of the very major errors were from a single site. And so, it's possible there were some testing discrepancies. We did go back and have essentially repeat the experiment with that site. And the very major errors stood. But 3 of them were for the same E. coli, were for E. coli. And so it's possible that that's just one weirdo strain of E. coli that this one particular site has. And this is something I think maybe to really highlight when you're doing your validation studies, is when you see those trends, it's really good to check into a couple of things. So the first thing I would always suggest people do is check to see, are there limitations for the test system that you have that the FDA imposed? So it was like a known issue. This happens all the time. Usually it's not for E. coli. And in this case, Microscan does not have a limitation for E. coli. So we kind of ruled that out. But it's a really good thing to kind of keep in mind, because you can't expect your validation to perform better than the FDA clinical trial. It's just, not going to happen. So that was the one thing. And then the other thing is, if it's all the same species, you might start asking yourself, did I just have really bad luck when I picked my isolates and I picked the one clone that is going to misbehave in the system? So we haven't sequenced these isolates. And so I think that's kind of an outstanding question with this. That being said, the major error rates were 4.8, which I think was pretty okay, and minor errors were pretty low, below that 10% threshold at 9. So, maybe slightly less than you would want. If this was my lab, I would keep working on this to just say, okay, let's see if we can resolve some of these discrepancies.
And then when we look at the VITEC, it performed really well. So 96% essential agreement, 95% categorical agreement. And similarly to Phoenix, it had one isolate in which there was a very major error. So again, that put this, because there were such small numbers in terms of your resistant isolates, it put that at above, that percentage we'd like to see. So, you kind of have to take that for what it is, right? So we would want to include more resistant isolates at that point. And no major errors, and only 2 minor errors. So again, compared to the broth microdilution, the VITEC system, it looked like it was performing really well in the hands of the lab that was performing the study.
I like hearing that. I mean, definitely that's the one that I work with. It's good hearing that it did well. So, with this question, definitely sometimes, a lot of medical lab scientists, they might not be aware of this. We're familiar with the CLSI, but then you see, FDA breakpoints, CLSI breakpoints, and some people might be completely, unaware about the breakpoints that the instruments have. And so how do the results compare, from your CLSI to the FDA? And also, while we're at that, why do both institutions, have different breakpoints? And how does it affect the automated instruments?
Yeah, so maybe I can talk a bit about the mess that is breakpoints in the US, because it is kind of messy. So the CLSI is a standard setting organization. So we don't have any regulatory authority. We just, based on the best available data through a consensus process, establish clinical breakpoints. The FDA, on the other hand, does have regulatory authority, and where they have regulatory authority is over the drug manufacturers as well as the device manufacturers. And so there's actually 2 different branches of the FDA that play those roles. So one is CDER, which is the Center for Drug Evaluation and Research. And so they have the drug side of it. And then there's CDRH, which is the Center for Devices and Radiological Health, which has the devices. And so by US law, devices like your VITEC, like your Microscan, like your Phoenix, they have to follow breakpoints that are recognized by CDER. So that's the drug branch. And so they publish these on a website. And typically those are breakpoints that are proposed to the FDA through the process of getting that drug approved for clinical use, for human use. There is a process that CLSI can use to pitch their breakpoints to the FDA, and we did do this for Piperacillin-Tazobactam. And so the FDA, for the most part, recognized the CLSI breakpoints this January. So it takes them a little bit, they're delayed close to about a year. I mean, to be fair, for the FDA, they've been dealing with this small pandemic issue on the side, so they've got a lot of stuff on their plate. But they did review the CLSI breakpoints, and for the most part, they did accept them for the Enterobacterales, with one small exception, which is that FDA doesn't really believe in SDD. They use intermediate. And so, but the numerical values are the same, so I don't think we need to fuss too much about that. And so now that FDA has recognized those CLSI PIP-TAZO breakpoints, the commercial systems can go back to the CDRH, the other branch of FDA, with their data to say, hey, our device works. And I know that all of these companies are working on that very hard, but you can imagine it's a lot of work. They sometimes have to redo a new clinical trial, and sometimes they have to redo some of their development if it's not quite formulated quite right. And so typically we see it can take anywhere from like 2 to 3 years all the way up to 10 plus years for them to do this. And this is kind of one of the unfortunate things that we're faced with right now in the United States, is some of our test systems we're kind of woefully behind in getting their breakpoints updated through the FDA, even when CLSI and FDA matched. Things are getting a lot better, but we did kind of go through this rough period, I'd say, over the last 10 years where there was a lot of disconnects. So I guess the take-home message I would say to labs is just because your device is FDA cleared does not mean that it's FDA cleared with the most current contemporary breakpoints. You really need to check with the manufacturer to see what breakpoints are being applied. And for those labs that are CAP accredited, that is now a CAP accreditation requirement.
Yeah, and to kind of piggyback on that, I would say this is probably a question. So Romney and I have been giving, workshops and we've done different things and talks about susceptibility testing. And this is probably, in my own experience as I, talk to people about susceptibility testing, that most people in the laboratory just aren't aware that that's the case, right? So you get this, M100, you get your update, you look at that, and it's just sort of common that you would assume that the device manufacturers, they come in and they update their software, that they're going to update your software to be the most recent or most updated breakpoints. And so it's just simply not the case in some of these scenarios. And so really that's something that in order to meet that CAP requirement or to do best practices within your laboratory, one of the things that's really recommended is that you kind of make a table for yourself, or you figure out what breakpoints you're actually using in your device, and you compare that with where CLSI sets, or go and meet with your stewardship partners or your pharmacists, your ID physicians, and have a discussion. What really should we be doing in this particular scenario? And then try to determine, is this something that we can update on our own? Is it something that maybe we think, if we speak with our device manufacturer, they're going to update within the next 6 months with that, that's coming out? Is there any assistance that we can get from our vendor to help us with that? I know some of these commercial AST systems, they're really looking at how they can help the customers, because they recognize not every laboratory is necessarily going to be able to do this on their own. And so they're really trying to figure out how to navigate the system as well, because they have this FDA process that they need to go through, but yet they're kind of stuck in the middle because they want to support their customers. So I think that's another piece of this, where if you're trying to figure out where to get started, that would be the advice that I give is just really investigate what breakpoints your system's using with your software, and then look at where you want to be. What are the differences that are current in terms of CLSI, and have that conversation, that multidisciplinary conversation, to figure out where you want to be in terms of some of these changes.
Yeah, and definitely I have seen out there, a lot of publications that now they're even, telling you, kind of giving you the steps of what to do, because, it can definitely be challenging, especially, where do I start? What do I do? So a lot of, sometimes, some labs even, they don't have, like a director on site or things like that. So whoever is, in charge of micro might not be too familiar with what to do. So it's really good that all this information is being brought out there.
And there will be, just to give a pitch for it, there will be a breakpoint implementation tool that is going to be available from the CLSI probably in the next month or two. And so that will have spreadsheets and how-tos and SOPs and all sorts of fabulous stuff to really help labs with this, because everybody struggles with it. So we get it. I struggle with it.
That's wonderful. So I will definitely be on the lookout for that, and so I can share it with the audience. So, with the study, what worked well, what didn't, and and then you mentioned, the very major errors. So maybe what do you think would be some of those possible causes?
Yeah, so I think going in, we sort of knew that the performance would be imperfect. We hoped that it would be really, really great, and it'd be like no big deal, and labs just update your breakpoints. But that's obviously not what we found. We did take a look at sort of the cost of doing nothing, right? So in our paper, we looked at CLSI breakpoints versus CLSI on the systems. We looked at FDA breakpoints versus FDA on the system, but then we looked at CLSI versus FDA. And that kind of painted, I think, the most important story of this whole thing. So if labs do nothing, looking across the systems, your very major error rate is between 6% and 45%. So that means just, sitting here today based on everything that we know about Piperacillin-Tazobactam, about how it's dosed, about the types of bugs we see today, if you don't make the breakpoint update, even though the systems didn't perform quite as well as we would have hoped, it's way worse if you do nothing. And so I think that was kind of like, at least for me, one of the most important points when we looked at this, because keep in mind, if you have an isolate with an MIC of 32, today that would be considered resistant. Yesterday, that was intermediate. And what we know from the MERINO trial is that result is associated with mortality for your patient if you treat them with Pip/Tazo. So I do think, despite how challenging this can be, labs really do need to make those updates. April, I don't know if you want to comment at all about the very major error reasons.
Yeah, I mean, I think when you go into something like this, obviously it's less than ideal to have one site that looks like an outlier, right? So, that can be problematic. And so whenever, let's say this is my site. So let's say that this happens, one of the things that I go into that looking at, if I was doing this validation in-house, is some of the points that Romney hit on earlier. Is this something to do with the particular isolates that I selected? So did I pick the right mix of bugs to begin with? And so often whenever we're doing these types of studies in our own institutions, we see that we don't want to necessarily overtax the system, right? So we don't want to pick all isolates that are right around the breakpoint. We want to have a really healthy spread of isolates. So ones that you would reasonably think the system would be able to call susceptible, and intermediate or susceptible dose dependent, or resistant. So you really want to have a spread. And we try to do that, honestly, in the study here. So that's one caveat again to this, where, you just may find yourself having a situation where you stumble upon a problem like this. And so when we've had in the past where we have set our criteria going into the validation or the verification, and we see that we're not meeting it. So either you can establish yourself potentially, a limitation, as Romney pointed out, or we say, hey, we're not really able to do that. But in this particular instance, E. coli versus Pip/Tazo, we would have to report that, right? We would have to figure out a way to work through that. So one of the things that we would want to look at is, is this a site issue? Do we have something that's going on at our particular site, where we're not setting these up properly? So we would assess our QC. We would look at other isolates. We would do some reproducibility, because in the clinical laboratory, unlike, in the research land, we don't do replicate testing, right? So you kind of get one bite at the apple whenever you're doing susceptibility testing on clinical isolates. So you have to be really sure that your system's performing in a way that you would expect it to. So again, we would kind of look at that and say, is it something to do with our system itself? So should we really thoroughly investigate our system? Is there something going on with either our panels or our instrument or our testing personnel, to really look and see if that was potentially the issue? And so if you're able to kind of reasonably rule those things out, then I think you have to go down the path of, is there something inherently different about this particular isolate or the set of isolates? And so just at our site, one of the things that we do see sometimes is that you have heterogeneity amongst, our E. coli isolates. So we do a lot of urine testing, I'll just say. We have a big healthy dose of outpatient clinics and outpatient testing sites or collection sites. So we do a lot of urine testing. And one of the questions that comes up pretty frequently is when we see that there's clonal variation amongst, E. coli or, an Enterobacter or a Pseudomonas aeruginosa, do we set up susceptibility on each one of those? When do we know whether to set it up, whether not to set it up, whether this is one thing, whether this is two things? So I think, it's perfectly reasonable to anticipate that maybe if you encounter some of these clinical isolates, you may have this phenomenon happen, right? And so if you're a laboratory that does disk diffusion testing, sometimes you can see intercolonies and you can pick those out. But when you're looking at, a broth plate, you're unlikely to be able to see that in the same way that you could with a disk diffusion. So I think, it's definitely possible that, it's an isolate issue. As Romney pointed out, whenever you have these resistance mechanisms, specifically ones that attack the beta lactams, you can have like an ESBL plus an AMP-C. These are already set near, for some of these isolates, near what would be considered susceptible for the wild type versus maybe inching over that MIC and now be in the resistance category. So really, we have to kind of think about how to interrogate that question. So that's kind of going to be next steps, I would think, for some of the laboratories that are interested in this to really look at, how do we better understand whether this is something to do with a phenomenon that we would see that may be reproducible somewhere else or outside this particular scenario.
Yeah, so definitely, my next question, you definitely touched on it and gave a great deal of information. So, I was going to ask about recommendations regarding the breakpoints, and you already talked about that. And, you also mentioned there's a tool coming and things, we can do. Is there anything else that you want to add about that? Or
Well, I would just say for labs that are CAP accredited, it's 2023 and FDA recognized the CLSI breakpoint for the most part. And so that means you have 2023, 2024, 2025. By 2026, you have to make the update per CAP regulation. They give you 3 years. So like I mentioned earlier, I know all of the commercial manufacturers are working on this. And it is, I truly believe, the better thing for patient care for us to do. I know breakpoints kind of feel a little esoteric sometimes to some of us, but data after data show that it really does make a huge difference on patient outcomes if you're using the most up-to-date clinical interpretations.
yeah, and that caught my eye as I was reading the article, because there's a section where it says, when you compare when the FDA breakpoints and then the CLSI breakpoints, some of the interpretations, change, right? And then the article says that some would have been reported one way, yeah, based on those changes. So
Yeah, for sure, especially for Pip/Tazo, right? Because so many bugs live around where the breakpoints are. Makes it trickier, but even more important, right? And I think it's, you talk about, as an example, carbapenem resistance. Most places that are, lucky, you're not seeing 10%, 20%. CRE in your lab, but you are seeing that kind of resistance rates with these new breakpoints for Pip/Tazo. And so, if you're not making the updates, that's a pretty big bucket of patients that you might be misdiagnosing.
Yeah, and I think the other thing I would add to that is, we didn't set this up to look at consecutive isolates, right? So if you wanted to really interrogate this for your own laboratory, you could pull out your antibiogram data. So our particular instrument allows you to get a distribution, like an MIC distribution. So then you can go back and look and say how many isolates or what percentage of isolates may fall in this category where I would be potentially not calling something resistant when it is. And so that would give you the ability to kind of in your own institution be able to make that decision as to whether or not this would be a problem for your particular location. But, to the statements earlier, I think, this is the crowd here that we're proponents of updating your breakpoints and really staying standard with what CLSI is putting out there, because those are really based on real world situations where we see patients are failing, we see that there's an issue. And so CLSI has really sought to try to correct and help laboratories solve some of those problems. And so we are, I don't want to speak for Romney, but I'm sure she feels this way, big proponents of really looking at how to get current with your susceptibility testing systems.
Absolutely.
okay, is there anything else you want to add?
No, other than to say, I think it was sort of a good case study in how tricky it can be to do these validations, right? And I think the other thing I would say is, as I mentioned, I know a lot of these companies are working on updating the breakpoints through FDA. They're going to have much bigger data sets, right? Like thousands of isolates, hundreds of isolates that they evaluate for that. And so I think the proof will be in that pudding, not this pudding so much. But this is more like a snapshot of what you're going to expect if you do this validation in your own lab.
Okay, well, definitely this has been, so informative. That's been great, and definitely a pleasure having you both here. So thank you so much for taking the time to coming into Let's Talk Micro.
Hey, it was our pleasure. It was fun.
Yep, thanks so much.
My pleasure. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about TCP and this study they did with, comparing the breakpoints and how the drug performed with automated instruments against a reference method. As always, I enjoy talking to you and bringing this information to you. Thank you for your support. Definitely, I appreciate it. Please continue downloading episodes and sharing the podcast. Thank you. And stay tuned. Great things are coming your way. As always, continue bringing that passion to what you do. That's very important. We have such a great work. We do such great work. As always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.